This video on homing endonucleases or meganucleases is largely a tribute to a genome editing technology from the 90s. Currently, we know of 4 to 5 major genome engineering technologies. They either involve chemical nucleases, meganucleases, zinc finger nucleases, talons, or everyone's favorite, the CRISPR-associated nucleases. Chemical nucleases are probably less relevant to this list, but meganucleases are important because they were used in some of the first genome editing attempts back in the early 90s. These attempts used naturally occurring homing endonucleases. The first such endonuclease was found in the light strand ribosome RNA gene in the mitochondria of yeast, which at that time was called omega, but now we know it as I-SceI1. Homing endonucleases are site-specific enzymes that tend to recognize large 14 to 40 base pairs of DNA sequences. And generally, there are 5 to 6 primary meganuclease families which are named by the amino acid residues that form the active site of each family. The lag family is the most widely studied meganuclease family. You may remember the HNH from Cas9, which has the HNH and RuVC catalytic domain. The catalytic folds in the HNH domain of Cas9 are similar to the HNH of meganucleases. This similarity is not surprising because all these enzymes are indeed endonucleases. Something to keep note is that these enzymes tend to be hidden as ORFs in group 1 or group 2 self-splicing introns. Sometimes even introns in I-CreI, and sometimes they exist as Inteins as well. And for this reason, when you see I-CreI as the meganuclease, the I means that this enzyme is found in an intron. Pi likewise would mean that it exists as a protein insert or an intein. And if you see F, it means that the ORF is in a free-floating context. Since they exist in broad context, we find them in mitochondria genome, chloroplast genome, spanning plants and yeast, bacteria, archaea, and even bacteriophages. Higher-order animals do not contain any meganucleases. In the rest of the kingdom, their stealthy existence in introns of proteins means that they generally evade harsh negative selection pressure. So you will often see that they have somewhat high evolutionary conservation. To be explicit, the intron-encoded endonuclease is not part of the original host protein. The host protein is spliced independently and the meganuclease is transcribed independently. This would be in contrast to an intein-type meganuclease, which is part of the coding host protein. When such RNA is translated, the intein excises itself into a functional endonuclease and the host protein matures into an independent protein. Generally, the homing endonuclease have no essential function to the host, and you will often hear about them in the context of selfish genetic elements. You will, however, find that they are involved in some processes like yeast mate switching, resistance against bacteriophages, and sometimes proteins in eukaryotes have co-opted meganuclease-like regions into their DNA binding domains. These elements generally expand themselves by the process of allele conversion. We can take the example of a diploid genome and suppose that one of the alleles has an endonuclease ORF, but the other allele does not. But it does contain a recognition site for the endonuclease. This recognition site then can be cut by the homing endonuclease, which then can be repaired using a homology repair mechanism where the ORF allele is used as the template. And this replaces the information, and you get an allelic expansion of the endonuclease ORF through copying. These 14 to 40 base pair long sequences are quite special, so let's discuss them a bit. I think it's more intuitive if we start with something simple, like restriction enzymes. For an 8 base pair cutting enzyme, you expect that a random DNA will contain 8 base pair site once every 65,000 bases. We have 3 billion haploid bases in our genome, which means we have roughly 46,000 sites for an 8-base cutting enzyme. But now, if you think about a 14-base endonuclease site, you expect to find only 11 sites in the human genome. For a 15-base, you have 3 expected sites. And if you go any higher, the probability becomes close to 0. So generally, you do not expect to find meganuclease sites in the human genome. One important thing, homing endonucleases are not restriction enzymes. Restriction enzymes have a restriction modification system, which is not part of the meganuclease function. Also, restriction enzymes, and I'm using EcoR1 as an example, it has a very specific DNA sequence to bind and cleave. In contrast, meganucleases rely on an indirect readout mechanism, which is typically done with the help of beta sheets within this enzyme. They can exist as monomers, dimers, or pseudomer proteins. So here's an example of a recognition sequence. It is not palindromic. It does have some inverted repeats, but they're not fixed in order. This site is recognized by a meganuclease called I-CreI. I-CreI is a dimer enzyme containing some alpha helices and beta sheets. Most meganucleases depend on some cofactors as well, like the magnesium ions. The beta sheets interact with the major groove of the recognition sequence, but they generally do not rely on a fixed set of nucleotide sequences. The recognition is largely part of a structural fit mechanism that ensures that the central 2 to 6 base pairs get recognized properly, that ensures the DNA cleavage. So the structural fit is sort of like a stabilizing mechanism, which ensures that in case of a suboptimal fit, the cofactor ions are ready to be kicked out. And this is called a cofactor displacement mechanism. But if the structural fit ends up being good, the central bases are then recognized without any problems, and the DNA is cleaved near the AT base pair that tends to leave about 2 to 4 nucleotides as overhang. So these stability and displacement mechanisms are quality control mechanisms to reach a proper energy balance. This also means that if you have some of these nucleotides mutated or mismatched, the enzyme might still be able to recognize the site and still cleave the DNA. And this type of flexibility does not exist in restriction enzymes. Okay, so how can we use them for genetic engineering? This was first shown in 1994 in Maria Jasin's lab. They created a mouse cell line containing an I-CreI recognition site with an antibiotic gene. Then you can transfect the cell line with an I-CreI enzyme expression vector and a donor fragment that you want to insert. Ideally, the donor is designed to contain homology regions around the meganuclease cut site, and it also contains a new antibiotic gene. This way, the donor can undergo homology-directed repair, and the final edited cells can be selected using the new antibiotic gene. This is the same idea as CRISPR-Cas9 system of targeted cleavage and HDR for knock-ins. In this case, the cleavage is done by the meganuclease. This method is, of course, not perfect. First, the fact that meganuclease sites are generally rare means that you first have to add them to the genome, which means you need to create a cell line that contains the antibiotic gene and an I-SceI site. Second, the I-SceI expression is not toxic to human or mouse cells, but other meganucleases, like PPO1 and CRE1, have some pseudosites in the human genome, so their expression typically kills the cells, meaning that the choice of meganuclease is also quite crucial in this method. Also, the meganucleases can tolerate some mismatches in their recognition DNA sequence, so it is hard to predict the sites that can still function as pseudosites, and that can result in off-targeting. People have found some workarounds to most of these issues. You can engineer existing meganucleases by varying specific amino acids in the enzymes so that it recognizes a new DNA sequence and perhaps with higher specificity. This usually means that you have some specific bases that you are trying to target as well, which means that the final recognition site is also in parallel engineered as the enzyme is also engineered. There are a few engineered variants that are used for cutting at specific locations in the human genome. Despite all this, their practical usage is low, even though they're compact, non-repetitive, and can easily be delivered using vectors or perhaps directly as proteins. Their limited usage is largely because of their unpredictable behavior in recognition site usage and potential toxicity. I hope this gives you reasonable familiarity with meganucleases. As always, I'll see you in the next video.